All right, so that should be pretty good. It's a little high. So the point of today was I was going to grab out my microscope. It's still a little loud. Grab my microscope and try and get some DNA on screen. So I have some preserved samples. I don't really know how well preserved they're gonna be, but I'm gonna try it nonetheless to try and see if we can grab out some DNA from them. Okay. So I'm gonna go to lowest magnification and I'll grab out one of these guys. So these are going to be fruit fly larvae that are preserved in ethanol. Click my slide into place. is really out of focus. There we go. Okay, so I'm not surprised, but this is just about as good as I'll be able to see with just this microscope. I don't think we'll be able to see much more through it. They're normally pretty clear, but these guys ended up turning a little opaque while they were being preserved. Yeah, it's not much better through the eyepiece. So it's not the camera, not necessarily. This microscope just wasn't made for something like this. So that also doesn't help. Changing the aperture doesn't really do anything. Yeah. Build that up. Yeah, so what we're looking at right here is about where the head should be. That's the head right there. If I can sort of, I don't want to move it around quite yet. But when I move it around, you might be able to see mouth hooks. So Drosophila or fruit fly larvae have little mouth hooks that are really hardened pieces of keratin that they use to dig through food and dirt and just plant material so they can eat up what they like to eat. And then it's just a worm, no legs, some segments that we can kind of see. Not well though, but I'm not surprised by that. And then just leading to the posterior end. So my camera is trying to autofocus, which is not helpful. Yeah. So the plan is to use these guys, and it might take a couple attempts. I'm not actually sure how well I'll be able to do this regardless. Oh, he's drying out. So these are in 100% ethanol. There. Actually, that cleared it up a little bit too. Yeah, these are in ethanol. So they might dry out a little bit when I add this rehydrating buffer to them. Yeah. It looks like something happened. So anyways, my plan is to take a couple shots at taking these guys, opening them up, and grabbing out a special organ called the salivary gland. These guys end up turning into pupa at some point during their development. Their salivary glands fill up with a sticky goo, and that's what they spit out so they can adhere to surfaces. So they can end up, adhering to like rocks or plants, or in the lab, they'll adhere to like the sides of tubes. So it'll probably take me a couple tries to get it. This microscope isn't a dissecting microscope, so it's going to be pretty tough to see what I'm doing. But I'll kind of take a guess at it. And also, I don't know what exactly preserved larvae are going to do. So in addition to that, I also have these living larvae in this tube I have at my desk. So these guys I'll be able to grab out fresh. They'll wiggle around, but we should be able to get something from them, even if I don't get anything from this. Okay, but I just kind of have to decide how to do this. Yeah, right now I'm at 4x plus 16x with the eyepieces. I kind of wish that I had something else like a 10x on the eyepiece for this, but I'll just grab out my forceps and give it a shot and see what we can do here. This is also going to be annoying because if I rest my hands on this microscope, it's going to change the level, which is going to completely change the magnification. And you know what? I'm going to have to try and take off some of these other objectives on this microscope so that my hands can actually fit. So I'm just going to remove the ones to the side really quick. Okay. And maybe what I'll do is I'll try to do it in a dish really quick. Or no, I'll try that later. I have a dish right here that I've had for a while. So that will let this thing stay in lots of liquid so it doesn't dry out as I'm trying to pull it apart. So let me quick just pull up a comparison so we can actually see what we're looking for. All right. None of these are great. That's fine. Yeah, so those are the mouth hooks that I mentioned. So those are up near the head. It has a mouth right here in the middle. It's missing its brain in this image, which is kind of right about here. This thing is mostly just a big digestive tube. So you can see the gut going through here. These Malpighian tubes are a type of tube to, I think, move around gases inside of the larva. The salivary glands are what I'm looking for, so that's right here. There's two on either side. They're really close to the mouth because they end up spitting out those glue substances out of their mouth as they are moving to the next stage of development. So what I'm going to try and do is I'm going to rip this thing in half, and then I'm actually going to push down from the top, kind of trying to invert the whole thing, so turn it inside out. And then if I can see them, I'll grab out those salivary glands. And then I'll immediately move them to a new slide, which I should probably prepare. And then I'll go from there. So I do have another slide right here ready to go. Yeah, we'll just try it, I guess. I'm going to lean over to try and look through the eyepiece. There's just enough lag with the camera that it's going to be really hard to see if I don't. Or it's going to be really hard to dissect if I don't do it live. So I kind of had to do that off camera just because this microscope is also inverting everything, so that's making it a little hard to coordinate. So I'm kind of looking outside of the eyepieces. But I've ripped the thing in half. It didn't completely collapse, so that's a good sign. So now I'll try and use my forceps to invert it, to turn it inside out. So I'm not doing a great job. I think these things are... The preservation might have kind of stiffened them up more than I'm used to. Also, as cruel as it might sound when they're alive and you kind of poke their heads, they kind of invert themselves a little bit, so then you just push them all the way through. Also, the microscope setup isn't helping, so it's a little bit of everything. Speaking of which, I think this is shifting a little. Yeah, absolutely. There we go. So I've pretty much destroyed this whole thing. I don't think this is going to work as well as I thought it might in this setup, but that's okay. So I'm just going to take this guy off. Just wipe it off. We'll try with a living one. Also, I'll try with the dissecting dish. I think this will actually hurt too, but might as well try. Ooh, yeah, no, never mind. That's not going to work at all. All right, so this will also need a little bit of liquid. I just have some PBS buffer. Just to kind of keep it wet. If I don't work fast, it's probably going to try and crawl away, but that's okay. There we go. So this one should have an obvious difference. So it's actually alive. Yeah. So the part we're looking at right now is its mouth hooks near its head. So it is. Let's just watch it. That's some of the food that it was just like stuck to it. Let's zoom out a little bit. Yeah. You actually see pretty well into it. So there are dark sacs on either side of its head that's bobbing in and out. When it extends, you can kind of see them. Those, I think, are the salivary glands when they're inside the larva. It's pretty dirty. Oh, you can see some tracheal tubes, so that's how it gets air. Yeah, so it's just trying to go around, find some food. It's got some more tracheal tubes up front. That's how it exchanges gases into its body. This darker part is that most of that's, fat and also the digestive system inside this guy. So he's not going to like it, but I am going to rip him in half. I'll move the half we don't want, which is the posterior end, out of the way, and I'll leave just this front part. He'll still try to keep on finding food and stuff with his mouth hooks that he's extending right there. You can sort of see them. There's also some strings that I accidentally picked up, so I'm going to try and move those out of the way too. Okay, so that is him split in half. A lot of his organs have started to come out. I hope I grow. Yep, that's the head. Great, I grabbed the right side. So coming out of it, you can actually see some more of those tubes and part of the gut. So now this is very hard without the microscope, and I'm not going to be able to use the microscope, and I'm going to try to invert it. So turn it inside out. Okay. Ooh, that was sort of successful. I'm gonna have to pull away some of this to kind of see what we've got. I think the salivary gland should be in there, but we'll see. All right, well, that looks like the mid-intestine. It's kind of like the right... it's been described to me as basically the stomach. So that's the thing on the left-hand side that looks kind of like a tube with tentacles coming out the top. I'm not actually seeing those salivary glands quite yet, so I'm going to keep doing some pulling. Hi, Paul. So what I'm looking at, actually, let me just switch quick to another screen. It won't go away. So I've got a fruit fly larva. So right now we're looking at kind of the upper half of it because I've cut it through this midsection and turned it inside out. So what I'm hoping to do today is get these organs called the salivary glands on either side. These have really, really, really big nuclei, so the DNA containing organelles inside of cells, and I'm hoping that we'll be able to actually see some chromosomes if I can get them out and squish them really, really tight with my thumb. We might be able to see some actual DNA. It's going okay. I don't have the right microscope, and I knew I didn't have the right microscope, so I'm having to do a lot of this by eye, and they're very small without the microscope. All right, so he's still moving around, but I'm going to try to clear away some of this stuff. And thanks for joining. So my ideas for my Sunday streams are more of a free-for-all, just kind of opening up for questions, doing things that I just want to do and try out. Like if this works, maybe sometime I'd bring it up again on the Wednesday night streams. So feel free to also just take me on a tangent if you want to talk about anything. This is just because I kind of had the time and the interest. Oh, actually, I picked out the brain. Hold on. All right, so in the center here, if I can move things around a little bit. All right. So let me bring up something really quick. This is also a test for some things. So let me open something really quick so I can actually draw on this slide. It doesn't help that he's still moving around a little bit. Okay. Why is it still moving around? Honestly, just because neurons are still probably firing. They're pretty simple. Also, my elbow is resting on the same table, so that's probably just my movement. So if I get my hands off the table, there, it stops shaking. What you're seeing is just like the small movements from me, like, you know, moving my forearm around to move the mouse. Because we're looking at this at about 4 times 16x. Oh, also it looks like I'm testing out having the chat on stream, and it looks like it's actually working. Okay, but now is this working? Is my question. No. Oh wait. Nope. Yes. Okay, awesome. Let's get rid of this. And of course that erases the background. Cool. Oh, no, it doesn't. Okay. So right about here is one organ, and then here it's kind of hidden now because it's moved around a little bit. This is the brain. Not at all what I'm looking for, but I found it. And this is something called a wing disc. So I'm just going to put wing. In the larvae are sacs of cells that eventually become the pieces of the full adult fly. So the wing disc kind of looks a little bit like this. It's got some swirly bits in it. And that eventually becomes the wing. The brain looks very phallic, and I don't understand the Twitch TOS enough to know whether or not I can actually draw it. It is 2 spheres and a cylinder. It looks ridiculous. It looks like a joke. So I'm not going to draw that, but I did pull out the brain, and actually these little tendrils coming off of it right here, those are individual neurons that are coming off of the brain. So again, not what I'm looking for. Oh, actually what I'm looking for, I think, is right here. That looks about right. Let me get rid of all this stuff. Whoops. Get rid of all this stuff if I, or not. There. Let me play with the zoom level a little bit. Yeah, actually that's the salivary gland right there. It's the sac that's kind of in the center of the screen at the bottom. So if I can just get that separated, then I'm good to go. keeps floating around a little bit, but there it is. It's okay if I get some of the extra detritus, because that will all be squished away. Let's see what else we got. We just have pieces of dead fly now, or dead larva. Ah, that little sphere in the center. My camera's moving a little. The little disc near the center of the screen. That's going to become a leg someday. Still got the brain right there at the bottom center, connected to the mouth. And then, oh yeah, okay, now I'm seeing where we are. Down here, that's some fat tissue. It looks a lot like human fat tissue, but that right there is exactly what I want. So I'm gonna try and grab that and take it off the slide and put it somewhere else. It's still attached to a bunch of stuff, and the camera's moving as I'm doing this, so I'll fix it. The camera's just my cell phone on a special mount, so I kind of have to work with what I'm given. So that's it. Looks like we have a brain still attached. Yeah, you can see the one of the two spheres at the bottom and the cylindrical part too. Oh, and mouth hooks. So part of its mouth right there, dead center. So that's how it eats. Okay, I'm going to pick this up and put it on a new slide though. Okay. This music is awful. Am I muted now? I'm still learning how to get this thing working properly. Yes, no. Okay. Clearly I just don't have my mic handled on that other one. So hello, Defested. Nice of you to join. Yeah, I'm not talking about Wraith today, but Paul, I was just going to say, I was talking to Defested offline, and he's got some really interesting ideas about the Wraith and things that I had not even considered because I was not prepared to go as far into the Wraith as we ended up going into. So I'll be getting back to those guys eventually. I think what I'm going to do is I'm going to make like a board with kind of, stuff we've talked about, stuff I want to talk about, and stuff I'm just going to avoid. The stuff that just I don't care about, doesn't make any sense. We can just ignore it. Anyways, Defested. I don't know how long you've been on, but I'm doing some dissections of fruit fly larvae, one of which is on screen right now, or a cartoon of it. I'm grabbing out a special organ that has a lot of DNA in it. I'm hoping that we can actually see some of that DNA with my microscope. Great, so I see you joined a couple minutes ago. We'll actually see if this works. So the reason I'm grabbing these specific organs is because if you've taken intro bio or bio in high school, you probably know that we have 2 copies of DNA in each of our cells. We have one that we got from our mom, one that we got from our dad, or the egg donor and sperm donor of your birth. These specific tissues in fruit flies are really, really interesting because they're what we call polyploid or polytene. That just means instead of 2 copies, they have hundreds or even thousands. And so because of that, we can actually see it under the microscope because they line up next to each other, making these really, really big, thick strands. Now, I don't actually know if I can do this with this microscope, but I'm going to try. So you're not going to be able to see what I'm doing, but that's okay. So I'm just adding the cover slip on, and now with some even pressure, I am going to push down with my thumb as hard as I can to break open the cells and pop out the DNA and get it to spread as much as possible. So I'm actually pushing down with as much force as I can for about 10 to 20 seconds. Okay, we'll see if that worked. All right. Scope camera. Let's move around and find some of those, see what they look like at a lower magnification. So all the blue bits are kind of dyed or stained pieces of fly larvae. Huh. I guess I really... Oh, there they are. Okay. That's what I was looking for. So not much. Oh, actually a really good look at those mouth hooks. So the mouth parts. Okay, so I'm just gonna zoom in and we'll see what we can see. Gotta clean up the top of my slide first. There we go. All right. Well, we're seeing high magnification of these mouth hooks. There they are. So this little bit right here, I didn't plan to have these on here, but they're fun to look at. This little bit right here, that's what's actually coming out of the mouth of the larvae. So they kind of go around scooping up, breaking up food. And the rest of the mouth hook is just kind of elsewhere in their body. So this part is like inside them, attached to muscle. Yeah, that's cool, but not actually what I'm looking for. So every time you're seeing some of this darker color, that is usually a cell's nucleus. Hello, Beyblade Forever. Thank you for following. Yeah, so all these darker bits are the cell's nucleus. Ooh, what is that? It's also very dirty, unfortunately. All right. So let's look at this one for example. We are looking at a fruit fly larva, specific organs, the salivary glands, which I've taken out and squished down really, really hard. And now I'm trying to stain them or dye them for DNA. So really hoping we'd see some chromosomes with this, but I think my setup, well, no, I know my setup isn't quite good enough for that, but we're trying regardless. Sorry, I'm futzing off screen with something. This is the first time I have this set up. It should be a way to annotate what we're looking at. You can see a little bit of red down there. That's because I wasn't able to completely erase what I was drawing before. But okay, let's make sure this is actually working. Okay, so, of course, it's on the wrong tool. Yeah, it is kind of neat. I'm hoping we're actually going to see something interesting. I've drawn a box there. Okay, so right here, that is the outline of the cell, and that is the nucleus. So the nucleus contains all of the genetic information of a cell. So this is one right here that we've found. If I can get my pen to work, just erase that. So these cells that we're looking for should be much, much bigger, way bigger than sort of the average cell. So I don't think we're seeing them quite yet. So I'm going to just zoom around a little bit more and see if we can find them. I'm almost wondering if these were it. This big mass of very dark blue. I'm seeing some individual nuclei in there, but it's not, I didn't squish them as well as I was hoping I was going to. This might take some off-stream practice. I might also just need to prepare some cells ahead of time. Oh, actually, so this really dark blue, I think these are actually, that was actually about what we were looking for, but they're just not dispersed enough. They're not spread out enough to actually see anything too useful, unfortunately. Shoot. Well, I can try again. It's just going to take a little bit. Also, my microscope is way dirtier than I thought it was after seeing this. You can kind of see the whatever is on the actual lenses is showing up really strongly here. Yeah, okay. I just gotta look in the eyepiece really quick after I center it. Yeah, so I think this is just some random staining. It's nothing really all that exciting. Let me I took off a piece of the microscope. Let me add it back. And that will let me zoom out just a tiny, tiny bit. Yeah. Let me do that. This microscope I have is a hand-me-down. It is no longer in production, and I can't buy parts for it anymore. My camera is just my cell phone strapped to the eyepiece, so it's all a little bit janky, but that's okay. It gets the job done. Beyblade, what brought you here this afternoon? Oh, yeah. Great. Yeah, so sometime soon. So on Wednesdays, my plan is to, through to Festin. Awesome. Yeah, my plan is generally to talk about just general biology or picking apart, in a good way, in a fun way, the... Oh, awesome. Generally pick apart like the good and the bad of some of the sci-fi that we see, bringing in an actual biologist perspective, and also through Defested, maybe someday more of a physics perspective. But at the moment, my Sundays are more just kind of free for all. I think this coming Wednesday, I was still planning on doing a Stargate stream. I wanted to talk a little bit about some wacky cloning that they have there. But eventually, so maybe not this week, but next week, I want to pick out something from Star Trek. Because there's a lot of interesting things going on in Star Trek, which are fun and fine, but also usually pretty wrong, but are kind of fun to theorize about. I'm having a very hard time finding... Oh. Here comes something. Wraith cloning or Carson cloning? Well, I was thinking more Asgard cloning. Those little freaks, I think have made some interesting choices in their lives. And I think I have some ideas for like why they kind of screwed up, because from one perspective you might think like, well, shouldn't they be able to figure it out? Like they should be able to like do better than us. We are currently undergoing two projects. This is great, by the way. We can talk about whatever you guys want. We, as humans in the real world, are undergoing two projects simultaneously. One is to bring back the passenger pigeon, which is an extinct species of pigeon that once was numbered in the billions here in North America. We killed them all for fun and to eat. They are really closely related to the, what is it called? Just the common pigeon, like the pigeon you see on streets of big cities. Defested says, especially since they found that one Asgard ancestor from pre-cloning Asgard. Yeah, exactly. So we are doing this where we have DNA from a dead animal and are trying to change the DNA of a living animal to create or de-extinct that first animal. Doing it with pigeons first, and we are actually actively working towards doing that with woolly mammoths. Like it's a real thing that's happening currently. And you know, if we dumb primates are attempting this prior to, you know, getting much past the moon, why aren't the Asgard able to really do much more? Well, I think I have some ideas for maybe how they found themselves kind of in that hole. And maybe it brings up some interesting lessons. So regardless, this is going on my hypothetical tack board that I'm going to make to kind of keep track of things to talk about. But yeah, Asgard cloning, number one on my shit list for cloners. They should be better. They should know better. All right, so I zoomed out a little bit just to take a better look at the material that we're working with. Oh wow, that's a really nice clear picture of those mouth hooks. Yeah, I may have destroyed those organs that I was looking for. Just like why they need humans to be the replicators, they are smart but not innovative. Yeah, that's kind of my my takeaway, which I think is actually really interesting writing. They're basically constantly hoisted by their own petards, and they don't seem to understand why. And it's potentially a consequence of their cloning efforts. All right, looking around, I'm pretty sure I might have just utterly destroyed the organs that I was hoping to get. I think if I try this again in the future with these guys, I think I might just have to do a little more prep work to get out the salivary glands beforehand, maybe mount them in some way before we go and look on this. Kind of like the Borg, not really coming up with new technology much, mostly just stealing others' innovations or the gold. But yeah, absolutely. Would the organs have gotten destroyed when you squished? Is that risk of squishing? Yes and no. So that's squishing with as much force as I could. You have to think about molecularly what we're doing. We're getting to the point where it's I'm trying to squish down DNA, which is really, really thin. There should be enough sort of space and force between the two glass plates, the cover slide and the cover slip and the slide itself, where I actually can't break the DNA. Rather, I destroy the entire cell, which is exactly what I wanted to do. But the DNA is squished down nice and safe. I think I kind of did a little bit of a middle ground where I think I wasn't able to squish hard enough. And I think I accidentally jostled it. So, up and down pressure, that's fine. That's squishing. But I think I accidentally moved my thumb a little, and that's shearing force. And that's going to rip apart everything. That is bad. That's what you don't want to do. And I think I kind of did it by accident. I do think some of these bigger cells that we were seeing up close to the mouth hooks, where I actually think there's more space, I think those were the salivary gland cells, but they just didn't pop open the way I needed them to. And out here in the middle of nowhere, I think it's just nothing, just destroyed tissue. Not sure what these stringy things are. That's okay. I have other things we can kind of look at. Oh, what are those? This isn't what I'm looking for, but there's some interesting group of tissue right there. Is this what I saw? Ooh. Oh, yeah. Okay. Sort of hard to tell. Yeah, I got to figure out a way to clean off these lenses. They're quite dirty. These are just small cells. I think these are fat cells, so they're much smaller than the one I pointed out before. The salivary glands and the other tissues in this area of the larva is usually covered in fat. So I think these, yeah, that's just fat tissue. I can just tell because they're smaller cells. So there's a bunch of cells right there. We can actually see better cells if I just take some from the inside of my mouth, because humans just have bigger cells than larvae of fruit flies. The other option is I have this nice little plate of goodies that I got this morning. So I have 2 mosses and some lichen. I wanted to just kind of, I don't know what we'd see, but I wanted to just sort of wash them off, take off the whatever comes off, and see if we see any little critters swimming around inside. So why don't I do that first? That's much easier. Because there's definitely stuff living in there. There is this gross pond down the street from me that I just felt weird going and taking a little bit of liquid from, but maybe I will in the future, just to kind of get something, because there's definitely going to be something swimming around in there. I'm also not a microbiologist. I do the next step down. Oh, what is going on here? I'm a molecular biologist, so we're going to see stuff, and I'm probably not going to know what it is. But that's part of the fun of just having a random microscope. All right, so I will rehydrate this lichen. I'm curious if there's anything living on the lichen itself, but it's kind of dry. I found it on my deck, so I'm just going to let that sit for a little bit. And then I'll wash off some of this moss that I have and just sort of pick up the liquid, and then we'll see if there's anything on it. Lichen. Definitely the second one. I'm trying to remember which one is which. I always hated that in the I don't know where that originated. I just assumed it was those So Bad They're Good movies, Underworld. Lichen. I just feel like someone in the room had to have said, like, hey, this is just like moss. Why don't we pick another word? And I get it. I know it's short for lycanthrope. Yeah, lycanthrop. I can't say it. Lycanthropy. Lycanthropy. That's how you put the emphasis on it. Yeah, but it's still dumb. You know, it's still a dumb name, which might be kind of genius. I guess if the vampires were just calling them lichens to be both accurate but also just kind of mean. Popularized by Underworld, but used before then. Makes sense. Oh. Okay, so this was just a first pass at some moss. I didn't really take up that much. It was pretty clear when I took it off, so I don't know if there will be anything on this guy. The magnification also could use some tweaking. That looks like something. I feel like if I take a dirtier sample, there would be more things in it. When I've done this other times, you can always see something swimming around. This might just be a fairly boring bit of moss that I found. I might, if I don't find anything very quickly, I'm just going to switch and get some more. And it's also always hard to tell. There's some, I'll just move it a little bit. There's some little dots that are spherical and clear. Those are actually probably just bacteria. Bacteria are pretty easy to find. I was hoping for something a little bit bigger. Maybe some water bears or something like that floating around. Water bears are supposed to live in moss, but they're also supposed to live everywhere. Kind of hoping, but I actually haven't seen one in person. Yeah. Tardigrades are very, very interesting. They are arthropods, so they're related to insects. And like arthropods and other animals like us, they use Hox genes to plan out their bodies. So Hox genes are these cool set of genes where it's like you'll have a gene that controls your head and a gene that controls your midsection. And I'm simplifying it super, like a lot to a large degree. But if you look at the, oh, there's something here, maybe. If I focus it might just be dust. Oh, look at that. Yeah, so tardigrades, they have Hox genes, but if you look at their Hox genes and compare them to those of other arthropods, like insects, tardigrades are just head. They just have head Hox genes. So they've evolved to get rid of everything else, but then they've just taken those head Hox genes and changed them a little bit to make them function as other parts of their body. So they still have legs. They still have like a head, like an anterior and a posterior. But if you look at their genetics, you'd think, oh, they're just all head. Yeah, so what do we got here? So this is some kind of single-celled, I'm just going to guess. I always forget the term. Plant, basically. Most of the time, if you're looking at something green in the type of sample that I'm looking at, you're probably looking at something that can do photosynthesis. But whether or not this is maybe actually some sort of bacteria, I don't know. That's cool. Yeah, I've found these guys before when I've done this. They also are pretty resistant to that blue staining. So what that could mean is that they are, in fact, maybe like much closer to a eukaryotic plant where they have a cell wall protecting the outside of them. Because I don't think they're bacteria. That's cool. Little guy right there. I'll stay in this plane and sort of zoom around. Because now I'm starting to see more as I'm focusing in and out. No one's swimming though. I was hoping to find something that swam. Oh yeah, here we go. So, oh, hello. Oh, hello. I found two things. So, okay, that bottom one doesn't seem to be doing anything. So I'll come back to him later. This guy, though. Ah, there he is. So first of all, you can sort of see some little dots swimming around it. Those are bacteria. Those are bacteria with either flagella or cilia kind of moving through the liquid. That guy is probably some sort of paramecium. I don't have the magnification or skills to tell you what species. So it's a single-celled eukaryote. Eukaryote? I think they're eukaryotes. Maybe don't quote me on that. Yeah, so he's swimming around looking for some prey. So they are predators. These guys are usually, I think they're usually green because of what they eat. Because they're eating little cyanobacter, so little photosynthetic bacteria. And usually they just stay green because of that. So he's moving around. I'll stop moving for a second so you can watch him swim very slowly. So they don't go by amoeboid movement, right? They're not moving their cell around. Instead, they have little swimmers, like little tails that kind of move them around in a medium. This guy, also very interesting. Very green. I don't see him, doesn't look like he's moving. I'm going to look under the eyepiece really quick. So unfortunately for you all, I have more clarity on the eyepiece. I just can't get the same with this camera. He looks nice and clear. I can see some structures inside of him, but I can't tell what's what. It's also not moving. It could really just be a single piece of the moss that I washed off, or he could actually be his own little thing. Let's move on and see. I'm really hoping one of these blobby guys will move eventually, and it will be like an amoeba or something. That's just detritus. Just more junk. That seems to be, those are some cells. Not sure what they are though. All right, there was a piece of moss on here that looked a lot like it had a lot more structure to it. So I might just take a bit of that, squish it down. We can look at some moss cells, and maybe there will be something that was hanging out on the moss too. Tardigrades are supposed to be everywhere, and yet I've never been able to find one. Not that I've looked too hard. What's this? Maybe that's just trash. Okay. I'm gonna grab off another sample. So at some point I'm going to get a Discord going, if nothing else, just to have a spot for suggestions. But I have just been dragging my feet on that so far. I've also got a chat bot going. But I'm still learning this on the fly. I don't understand half of what all the stuff you're supposed to do for a proper stream. All right, this is also going to be a bad one, but might as well try. Oh yeah, absolutely. I mean, careful to open yourself up for me asking for help, because then I will constantly. But it's even simple things like me not knowing that I was muted on that one source that I had, or not source, that scene that I had set up in OBS Studio. Why isn't there a warning that says, hey, you know, this is a kind of a quiet scene. Did you mean that to happen? Also, whether or not my Fossa bot notifications are silent, or if they tell everyone what the person, you know, the command that was just run. All right. I normally tell everyone. All right, well. Oh, here we go. Paul, I have to go, but thank you for this incredible stream. Always been a literature and writing student, so this is all new to me, but this is also interesting. You teach well. Well, thank you very much. Yeah, you know, continue to join if you can. I will try to be diligent about saying what we're talking about on Wednesday nights, but Sunday afternoons are just supposed to be for whatever. This, talking, me working on slides. We'll see. But thanks for coming. Have a good rest of your day. All right, so this is like a little nodule that I found on top of this moss that I had. I just kind of picked it off just so we can kind of see if anything was hanging out on top of here. If not, I'll be a little bit more aggressive and grab some of the dirt at the bottom. Let's go all the way in. Ooh, that's... Oh, there we go. Wow, very cool. So moss, I think, is what's called a non-vascular plant. Slob. I use Streamlabs OBS. If you are using OBS Studio, I'd be a lot less familiar with it. Yeah, I'm using OBS Studio. That was what was recommended to me by another streamer who I've been watching for quite a while. And to be honest, I like OBS Studio better. I did both at first. Like, I downloaded both, opened them both up. Just wasn't a huge fan of Streamlabs OBS, but I will use Streamlabs like for their widgets and stuff, which are all compatible with OBS Studio. It's just I didn't like it felt like there was less customization. Maybe I just had to learn better, but at the time, I just felt like using OBS Studio. But all the widgets and stuff are completely cross-compatible. So okay. So anyways, yeah, I think moss is a non-vascular plant, so that means it doesn't have vasculature, unsurprisingly. So that's why they're always so small and low to the ground, and usually in a wet area. It's because they don't have a great way of transporting water throughout themselves. So whatever I picked up had these little spheres, little green spheres on top, and that's what I just picked up and squashed down. So whatever those spheres are, I don't know if they're a reproductive organ of some kind. We're looking at the outside of them, so it's clearly some kind of like outer layer. The vein, what look like veins, are probably the cell walls of the individual cells inside, and we can actually see pretty well inside of these, and there's some structure. I'm just going to look under the eyepiece really quick. Actually, I moved the camera. Actually, this time it looks a lot better on the camera, not the eyepiece. There's a lot of background that the camera's not picking up, which is nice. But these little dots, little things inside of each of these cells, those look real. I'm not quite sure what they are. They don't look like chloroplasts necessarily. These cells are green, so they probably have chloroplasts. But it might have just been from me squishing it that it looks a little off. I decided to squish rather than try to take like a section. But I do know on the periphery there was some other just cell debris, so that's what I'm going to look at really quick. Yeah, it just looks like cell stuff. Nothing too exciting. Let's go to the end. This is what I wasn't sure about, whether or not there'd be any little guys hanging off on the outside. Not much. That's less interesting. Let's go back up here. This is just really pretty. I don't know very much about plants, but I just think they're really interesting. Yeah. So I am just going to pause on this guy. I'm just going to pause on this, and then I'm going to just sort of like agitate and suck up some of the liquid. Oh, actually, here, I'll take some from the lichen, which again, I don't know if there's actually anything in it. It was very dry. maybe if there were things living on it, they needed to sort of rehydrate for a longer time than I'm going to give them. But we might as well check. I guess another thing, if I actually started Discord, like I say I will, I could take suggestions for the microscope. And if it's not too gross, or actually I don't know what would be against term TOS for a microscope. I can guess, but I don't actually know if they have specific language about certain things. I can get suggestions for what to actually like look at. The lichen actually came somewhat back to life after I added some water to it. It's looking a lot more green and a lot more fresh. So good for it, I guess. Okay. Get this off. If you could get some wraith cells, it'd be cool to look at that. You know, I really wish. I don't think so. But I was thinking one cool thing could be just sort of a screenshot roundup. So they always are displaying something off, you know, in the corner on one of their screens. You know, some like medical information or something like that. Or they'll have pictures of cells or DNA. And if you pause and look, it's usually pretty funny because it's usually pretty wrong. Or like you wouldn't look at DNA. There we go. Am I actually looking at what I'm thinking? Yeah, basically, oh yeah, anytime computer code is shown, or something I'm sure that, you know, people have done for the various, you know, shows or properties where there's a language translation available, you know, going through and translating all the random stuff that they show on screen in alien languages. Like I know in Stargate, the Atlantean script usually says something in English. All right, this lichen might be... I'll zoom in, but this lichen might be a bust. I don't think anyone's home. Yeah. I don't know what this was or is. I bet all this stuff was alive at one point. And I wouldn't be surprised if some of those dots we're seeing in the background are living organisms. You know, they're probably like fungi or bacteria. But if they're not big enough to see clearly, I kind of find them boring. Yeah, that's not really much. Oh. I mean, yeah. All right, I'm officially bored of this lichen. I'm gonna grab some dirt. It's been really warm and rainy in my region, so I'm kind of hoping that things have been living in the soil. Now I'm really regretting not grabbing some of that dirty pond water that's down the street. But the way it's it's in an area that's taken care of by the HOA that we don't pay for because we're renters, and everyone's always walking their dog down there. I would just feel weird as just an adult going down there with a little pipette, sucking up a tiny bit of water, putting it in a tube, then that tube in my pocket, then walking away. But if it's interesting, maybe I should just go ahead and do it. No one can stop me. All right, so this is turning out to be a different color. It's probably just from the soil. And at least in this area, the color of a lot of the soil comes from the tannins released by the decaying plant matter. So we'll see. We'll see what this looks like. Whoops. Oh. Well, it's dirt. That's for sure. Oh, there is something right there, though. Right away, one of those rod-shaped guys. Pausing to see if I see any movement. Not quite. So this is not the highest power I can go. Oh, there's something there, too. Yeah, I'm seeing... It's easier to see the ones that are green. Now, I could use that blue-colored stain that I have. That stains for... basically DNA. Only problem is that just kills everything. All right. That rod looking thing also might be something that's alive. If I kill everything, it's far less interesting to watch if it actually starts to move around. I'm going to zoom in. All right, well, that rod-shaped thing is just a piece of dirt. But at least there's more stuff to investigate here. I am sort of shocked by how dead all of this stuff is. You hear that like water bears, like they live on moss and they're so easy to find and they're everywhere. Not to me. Oh, hey, that's something. What are you? Oh, wow. Okay, so this one, it's clearly photosynthetic, and if I sort of go in at about that level, you can see spherical shaped things. Let me just draw more what I'm seeing. Nope. So looks about like that. There are some spherical speckles in here of different sizes. What you can't really see from this camera is that these, oh, don't know why everything just shifted. They are a much more specific, it's hard to describe. They are a different green color. Like this is sort of like a sickly yellow green. That looks like it's just the cytoplasm, the inside of the cells. I don't know necessarily why it's that green necessarily, but inside these spheres, it's a much more bright green. To the point where I actually wonder if the color filter that I have on this is making it harder to see on the stream, but not on my camera itself, or not the eyepiece. Anyways, I think those might be chloroplasts. So those are the actual photosynthetic organelles. Those are the things that take sunlight and derive energy from the sunlight, which is how things that do photosynthesis actually get energy, right? They take in, or they harvest the energy from the light, I should say that. They harvest the energy, and they use that in order to turn carbon dioxide and water. Or no, not carbon dioxide and water. Carbon dioxide and why am I blanking on this? So carbon dioxide and water, right? Carbon dioxide and water in order to harvest it, harvest the energy. They make ATP out of it, then they can store that ATP in carbon-containing compounds. It's mixing up my cellular respirations. So that little guy is kind of cool. He's very big, too. I don't know if they know. Oh, that might be another one. Oh. It's got kind of a different structure, but similar thing. Probably photosynthetic. Those are going to be the easiest ones to find in this, because some of these blobs, they very easily could be living cells, but if they're not moving, it's kind of hard to tell. Like here we have kind of a group of things that have sort of an amoeba shape, in that they don't have quite a defined outside, but they have that sort of coloration. And a printer is going off in my room. So, sorry for that. There's that guy again. No one's moving though. That's sort of frustrating. Oh. Here's another one. Same species. So I wonder if that other one was in the act of dividing. This one seems a lot longer, and you can kind of see, if I zoom in and out, this sort of cell wall looking structure seems to be separating two different cytoplasms more completely than the other one. So I wonder if these guys are undergoing mitosis or cell division. By these guys, I mean these two, and then the other two. Because the other one also kind of looked kind of long, and maybe like it was sort of separating, but I didn't want to say anything necessarily because I wasn't sure. But looking at that makes me think that they actually are. This is going to get really hot under the lights, so I bet these guys would die if we watched them for too long. But it'd be kind of cool to get a time lapse of something like that someday. I'll have to think about how to do that. Since most of this is just dirt, it's going to be cell debris, bits of insects, bits of plant. Also just inorganic things like pieces of sand. There's another, right in the middle there, there's another one of those cells that we saw before. Oh, hello. So that clear rod-shaped thing, I'm just pausing on it to see if it's going to move. Whenever I see sort of like a, not rod shape, like an elongated oval or an ellipse, that always makes me think paramecium. But it doesn't seem to be moving. It could still be, it's just either dead or just deciding not to move. Huh, yeah, a little disappointing. We're seeing a lot of cells. We're probably seeing more cells than I'm pointing out just because I can't identify things that aren't really plainly obvious at this magnification. So those are things that are usually photosynthetic, so they're very obviously green, or things that are moving around under their own power. So if we look very carefully, that's going to be bacteria, but those are going to be way too small. But those paramecium are pretty easy to see when we do see them. More amoeba looking guys, but they're not really moving. So given the time, I've scheduled myself to have 2 hours for this stream. Oh, wait, hold on. What are you? Can't quite get him in focus, but there is something moving around right there. Oh, just missed him. That size, it could... Oh, there he is. I think that is a very small... I'm going to say eukaryote. As he comes in and out of plane, you can kind of tell that there's like little dots inside of him. Actually, I guess I can't make that guess. That's something. So I'm not sure what that is, but it's pretty big for a bacteria. But then again, I'm not a microbiologist, so... Let's move on. Anyways, given the time, I might just do one last thing, which is swab my cheek for cells. Those are really easy to see. Those are nice and big. They will be dead. But you can really see the nuclei on those. And speaking of which, so those guys right there, I just know that won't be affected by my stain, but those two center things, I doubt those could be the nuclei, but plant cells often have something called a centrovacuole, which is a large, basically sac on the inside of them that they use to store materials, usually like starches, things like that. And often you can see them very easily. So I'm kind of guessing that's what those are. Yeah. The point of the stream was to find some DNA, which we kind of saw in those nuclei. But I'll have to try again off stream to see if I can get some actual chromosomes on camera. Okay. I'll leave it on this while I just prepare one last slide with some of my own cheek cells. If I can find my slides, there they are. So, human skin cells on the inside of your cheek are just really nice. They are generally pretty flat. They're pretty big, and it doesn't hurt to obtain them. It'd be kind of cool to get a blood sample at some point, but I don't really want to take blood from myself. I need to find a way to just more or less safely give myself a pinprick to look at that. And blood's nice because you don't need a stain to see the red blood cells. They are pretty obvious. White blood cells you can sort of use a stain to look at. Do I know someone with diabetes? Yes, but not in my area. Also, I'd want it to be pretty fresh. So, you know, sitting right here is kind of where I'd want it to get collected. I don't want to bother with trying to preserve cells. All of this so far, except for earlier before you got on, when I was trying to figure out some preserved fly larvae that I have, which didn't really work out that well. That's just a whole other layer. They have the little needles that are nearly painless. Oh yeah, that's true. Just take some of their diabetes supplies. I'll think about that, but that is a good idea. If I can think of someone who has diabetes that I know in the area, I can get them from very cheap from pharmacies. You can without a prescription for diabetic materials and testing supplies. I mean, I guess it doesn't hurt to ask. Just looking around for my little scraper. So primarily what I just use... Oh. Slayer Darth, sorry. Thank you for following. You don't need needles. I don't think you need the needle, or the needles need a script. Insulin definitely does. Yes, absolutely, definitely does. I'll look into that. That would be useful. But for now, my super sophisticated medical equipment is a zip tie. It's just nice because they are easily cleanable and they are just a stiff piece of plastic. Lancets. Oh. All right, I am going to write that down. Yeah. So you just kind of roughly swab the inside of your cheek. People do this in high school all the time. So hopefully I was gentle enough not to have destroyed all of the cells. So they are on my little glass plate. And now I will drop just a little bit of my blue stain on them. Hi there, Dr. NPHD and Defested. Hope you're both doing well. Well, thank you. I hope you're doing well too. Oh, awesome. You did kind of catch the very end of today's just sort of general stream, but we've been playing around with my microscope, trying to find something interesting to look at, basically with dirt and moss that I found in my backyard. Next time, I'll get some liquid from the gross pond that's just down the street. But right now I'm preparing a slide of my own cheek cells, which will hopefully be a little bit more interesting because you can really tell what the cell looks like. Do you have any interest in sci-fi? My other planned group of streams on Wednesdays are going to be sort of like looks into the science behind or suggested by science fiction media. I used to just what was expected of me in high school. Oh, I see. Yeah, so I think I love sci-fi, especially bad sci-fi and good sci-fi. A lot of them get things surprisingly okay, and a lot more things surprisingly wrong. Now that I'm older, but not necessarily wiser, I have a much deeper appreciation for a lot of subjects. Awesome. Well, if you're interested, come check that out on Wednesdays. This week, I'll be talking about a very specific property, Stargate, and talking about a group of aliens known as the Asgard and their dumb, stupid mistakes that they did while trying to clone themselves. And in that, we'll talk about cloning, like what cloning is, how you could do it, how we have done it, and how I think these silly little aliens should have maybe known better. Granted, of course, knowing that it's all, they're all made up. Yeah, so actually, if you wouldn't mind trying something, type !CV. I have a bot that should say what I do, but I forgot how much information I added to that. Hey, there we go. Look at that. Nice to know that worked. Yeah, so I'm a molecular biologist, so I don't study the cells like what we're looking at here, which I scraped a lot. I study the molecules inside of cells, so proteins and DNA are kind of my focus. Currently following 2 evolutionary biology streamers. There's a... Let's see. I was following, what's his name? Evo Lizzie or something like that. Very interesting. My undergraduate had a focus. Jack's Facts. I should write that down. My undergrad had a focus in evolution, but I was more a biology person. But nowadays, I'm very molecular. I like things that are less theoretical, more practical, and we do a lot of biotechnology type things. CRISPR all day long, basically. Which is another subject that I will talk about in the future. I think we're all very molecular, TBH. Yeah, probably in more ways than one. Oh, wow. Here we go. Here's some cells. Nice. Okay. Oh, wow. These spread really nicely. Okay, this is beautiful. So, what we're looking at are just epithelial cells. So these are from the inside of my mouth. Science streams. Oh, yeah. I know them via another streamer. Not personally, but I know of them. Okay, what's your question? As you're typing out, I will point out that this darker space in the middle, that is the nucleus. That's exactly what I was hoping to see. So if you remember, well, it's hard to say, or it's hard to maybe remember, but when I was back on that Drosophila larvae, the cells were much, much smaller. Human cells are huge. Okay, why do dogs and cats with albinism have a higher chance of being deaf? That's interesting. So I don't know the genetics of albinism. The basics, though, are clearly that they don't have... Oh, it's because the cells for determining pigment color comes from the same stem cells for hearing, right? See, I don't know. That's a good question. I can look into that. But cells containing melanin are that's the cells that confer pigment. IDK how all the genetics works, though. Sure. Let's hear. I was planning on ending soon, but honestly, this is an interesting enough question that I will look it up right now and we'll talk about it. So, good question. Let me just reorient myself. Set them away from the microscope. Yeah, so there is this word. It's the word is pleiotropy. Oh, by the way, what? Please share away. Core ACD. World's largest collection of open access research papers. This is a good resource to have in my back pocket. I actually have, I have been here previously. I probably should use it more just to support it, but I work at a university, so generally I can just get papers regardless because we have a university subscription to everything, but supporting open access is generally a good idea. And this is a confusing subject because in some ways I think, and I am totally okay with being wrong, that in some ways I think you do need a little bit of a barrier for publishing. But that said, the barrier we have is way too high. Everyone should be able to see the research articles that are federally funded and published. It is currently not like that, and that is wrong. But also, in order to submit papers for review to certain journals, you have to pay quite a bit of money. I think that's okay, but that quite a bit of money should probably be way less. Okay. So I don't use JSTOR. So we just have individual subscriptions, but to hundreds or thousands of research journals. So I can effectively go to scholar.google.com, search any article, and I have access to it. Okay. But let's look up this. But this is interesting. You brought up albinism. Genetics. So there's this thing called pleiotropy, and that word sort of describes in genetics. So when you have a gene, it is affected in some way, and by affecting that gene, you have downstream effects that are maybe obvious once you do a lot of research into them, but maybe aren't clear initially. So pleiotropic effects basically describes how biology is not just a series of genes in a series of vacuums that are all doing only their one jobs. They're cross-talking, they're interacting and having emergent properties that are maybe not predictable by just knowing the function of each of them separately. And so I hadn't heard this about albinism, but I'm sure that albinism is probably going to have some more complicated effects than simply not having melanin. Most research papers are written for other scientists. That is not necessarily a bad thing. Scientists should be part of a broader sort of machine of interpreters and peer editors. So you need to be very clear as a scientist in your research papers and very open about what you've done and the results you get and your conclusions that come from them. And so because of that, you do lose the public-facing sort of aspect of it, which is why always, like every 3 years, we get a newscaster somewhere saying like, "Oh, the cure for cancer has been discovered." Well, that's not ever true, but that's not ever completely false. It's like they just don't understand quite how what am I trying to say? How granular science gets, and we are slowly moving it forward. And then you have people like scientific communicators who are sometimes scientists, sometimes not, who are able to sort of take information and interpret it for the public. That said, there is sort of a shift in thinking about it. A lot of people who are writing their theses, so their master's or PhD theses, have started to include public-facing chapters. So a chapter that is free of jargon, free of complex ideas, but gets the point across, but without like being like rude to a layperson. You know, bringing you along for the journey, basically. Yeah, that is something I was going to ask you about offline because I became victim to one of those recently, and I'm still unclear of what it means. Okay. Sorry, I keep moving that around. So, okay, so you say albinism in cats and dogs, right? Is there something you could point me to? Because I don't know. I thought humans also, if you had albinism, you also had problems, not just with eyesight, but that makes sense, but also with hearing. Let's go to good old medlineplus.gov. Just gonna read this. That's not helpful. I'm already just skimming what we just like zoomed by. Oh yeah. In mice, a total of 100 genes are known to affect albinism. Yeah, so already, that's not a good sign for me giving you an answer on this stream. So this is clearly going to be a really complicated disease. So some diseases, right, it's one gene, one disease, and it's pretty straightforward. Other diseases are not that straightforward. Just cancer as a broad category, which we think of cancer as one disease, but it's many individual diseases in some ways, not in others, is an example. Clearly, albinism is going to be. But I see that you've sent me some links, so let me just open these. heritability and complex segregation analysis of deafness in Jack Russell Terriers. Oh, that's fun. Endogenous retrovirus insertion in KIT oncogene determines white and white spotting in domestic cats. Okay. How do albino fish hear? Okay. So let's start with this one. Endogenous retrovirus insertion determines white spotting. Yeah, so let's see. Segregation and sequence analysis of KIT gene in 2 pedigrees, P1, P2, revealed the remarkable retrotransposition and evolution of a feline endogenous retrovirus, or FERV1, as responsible for 2 distinct phenotypes of the W locus, the dominant white, and white spotting. A full-length FERV1 element is associated with white spotting, whereas a FERV1 long-terminal repeat is associated with all dominant white individuals. Interesting. So okay, so this already shows me something. So this is going to be completely different than other types of albinism. What this is already describing, an endogenous retrovirus. This is a DNA invader element. So it is very similar to like HIV. HIV is a virus that humans can be infected with. HIV contains RNA inside of that virus. That viral RNA is reverse transcribed, meaning it's turned into DNA and can be inserted into the human genome. I'm forgetting my HIV biology, so I'm not sure if it's actually inserted into the genome. A virus will replicate by infecting a protein cell, whereas a retrovirus will load RNA. Yeah, so all cells make protein, first of all. Viruses work by a huge variety of mechanisms, generally by injecting their genome, so their DNA or their RNA, into a host cell, taking over some amount of host cell machinery and using that to their advantage. And that advantage is making more viruses. So some viruses insert their genome, they get the cell to make their capsid, so like the proteins on the outside of them, they get assembled and they explode out of the cell. Like literally they just fill up the cell until they explode. Other ones leave the cell, stealing a bit of the cell's cell membrane as they go, also ultimately ending up in their death. So you are right. You are on the right track. Retroviruses, yes, they load RNA, and the retro, I think, I'm not a virologist, but I think that means the retrotranscription. So they are usually taking that RNA genome, making it into DNA. I think sometimes that is inserted into the host. Now, sometimes we have something that's even less than a virus. It is something that doesn't really contain its own proteins anymore. Maybe it used to come from a virus long ago, evolutionarily speaking. But what these are pieces of rogue DNA, or sometimes RNA, that hop around and live inside of the genome of an organism. So is a retrovirus's aim not to replicate itself into copies, just to modify the host DNA? No, that is its aim, but how it gets there is by modifying the host's DNA. So why don't I draw this out? Give me a second. Actually, let me hide this quick so I can open up my files. Very good questions, though, by the way. Okay. So, is this going to work? So the host has DNA. Easy, right? This is the host DNA. Oh no, you don't have to surprise me with questions. That's fine. This is not usually how we necessarily denote it, but I am going to denote proteins, or genes rather, this way. I don't want to fully rely on a person of whom I'm asking the questions. Should be bringing something to the table. I mean, it seems like you've done some research, so I appreciate that. Anyways, so these are different genes, right? And this will actually get back to the cat thing. So DNA invaders, right? They can exist. They are either viruses, part of the virus life cycle, or honestly, they can just be free floating, right? So let's just say we have a DNA invader who is existing as an RNA. Maybe that came from a virus. Maybe that is what is called endogenous, meaning it actually was already in the DNA. We have a lot of those as humans. I think all eukaryotes have them. Specific structures of our DNA, of our genomes, actually originated as DNA invaders who kind of just broke, but their DNA skeletons have been left behind. So anyways, in our virus example and our cat example, we have this RNA, right? And this RNA has a very specific function. This RNA is the DNA invader. And what does it want to do? It wants to survive and reproduce or replicate, as you've pointed out. Now, the way an RNA, a retrotransposed or retroelement invader does this is by inserting itself into host DNA. Sometimes that happens in what's called intergenic space. It is important, but it's not a gene. It's not breaking something necessarily. So what you would get if you did this, I don't think I can actually break this up. I'm not going to redraw the whole thing, but you might get, you get your genome, get like that, and I am going to color it red, but now the invader is in the form of DNA. But suddenly you get this, right? The DNA invader is now in the genome of the host. I'll draw a couple of these genes. And if you think about evolution, the reason why this is really advantageous is because now this little invader can stick along for the ride, right? It's now survived to the next generation because it's in a very permanent form. And now this guy might eventually hop out and go move somewhere else. Happens all the time. Another way that this guy can sort of cause trouble is it can insert itself into a gene. So like if this gene, for example, was one of the causative genes for albinism, so maybe it's important for making melanin or important for the survival of cells who have a lot of melanin. Suddenly, if I draw out the resulting genome, get this. Now if we draw where that gene is, now we've got a problem. I'll draw this other gene to just illustrate. I should point out these arrows kind of are denoting like, oh, it starts here and goes that way. Well, now this guy has a big interruption. So it's not like an arrow. We can just put a little X. Yeah, this isn't even, I would call stuff like this molecular genetics even, so it's even a thing further removed. Okay, so now you've got this little RNA or DNA invader, in the middle of a gene. So whereas this gene up here, let me pick yet another color. Whereas this gene up here will go and make its own RNA, and that RNA will become a protein. This guy, it might make RNA, but if you make a protein with that big thing in the middle, it's probably broken. So I'm just going to summarize and say no protein. Yeah, so the process of making, also, hello, Blamin16? Yes, messenger RNA. So when we're talking about, oh, I see. Yeah. Funny thing is, I feel like the more in depth you go into science, you stop saying mRNA, tRNA, rRNA. You just say RNA because everyone knows which RNA you're talking about. So that's my fault. Yes, this was mRNA. This mRNA that's resultant usually won't make a functional protein. So for the cat example, if this protein was like the melanin gene, well, now you don't have it. And so this paper that you've provided, from my very, very brief skim of the abstract, seems like they've found that a retrovirus is inserting, that's what we just saw, into this particular gene. Now, I don't know why it's an oncogene yet, so that means it must be related to cancer in some way. So perhaps by that inserting, you're getting two things: increased risk for cancer and a white cat. Now, the endogenous part is interesting. The endogenous means it was already within the environment. So in terms of what I've drawn, it might have been these first two cases, right? So endogenous means it might have been here. Thank you for the follow. Oh, that's a difficult name. Gretsiak? Thank you for the follow, regardless. So you might start here at number 1, where endogenous, that this retroviral, or this retro element is just hanging out, not causing any problems. It's just kind of along for the ride. And it's getting replicated, so it's happy. But one day it decides to move, and just by chance, maybe it's more likely to move into this other gene more than other places. So in step 2, asked science a few days ago about why things have non-functional genes. Pretty much got this explanation almost exactly. That's good, I guess. I don't watch them, but I think they're also scientists, so I'm not surprised. Also, I'm glad I'm giving a good explanation. I wasn't expecting this line of talking today, so great. I'm glad I wasn't given too hard of a question. Anyways, retroviruses moving around, retro elements moving around, pretty common thing, happens in all of us. Things have certain proportions of occurring. So there are small elements on the ends of this little red guy that maybe make it more likely to pop in and out of certain places. Sounds like in cats, it's really likely to show up in this KIT gene, which is kind of a fun name for a gene for a cat. And not only is this KIT gene an oncogene, meaning it is related in some way to cancer, and by screwing it up, you're probably giving a cat cancer or giving it a higher likelihood of having cancer. You are also giving it some form of albinism. Yeah. I hope that explanation did that help a little bit? I see that you say Slayer Darth. I see that you're saying, oh, I see. So I hope that means that I've sufficiently answered your question. Let's go back to here. Yeah, this is interesting. Let's see. Retrotransposon interrupts a DNase 1 hypersensitive site. So that just means that it is not protected by proteins in a way. So that makes it more likely. I see. Yeah, I think I've got a better understanding. Thanks. Awesome. Oh, it's highly conserved in mammals. Demonstrated to regulate a temporal and tissue-specific expression of KIT in murine, so that's mice, hematopoietic and melanocytic cells. So that's where you get some albinism. If you're starting to kill off these cells or never grow them, you're not going to have a lot of the melanin. I'm a layperson at the end of the day, so I'll recatch parts of the explanation that I need to refresh on and probably do some more reading. Oh, good on you. Yeah, keeping yourself informed is really important, also really hard. So good job. That doesn't mean I know why it's in. By the way, what's great about Core is that you can filter search results by fields such as biology. Yeah, that's pretty common for different types of journal aggregators. But this is a good resource to know about. I will definitely read this more, or look into this more, and probably use it more. It's probably good to support like an open access group rather than just Google. I'm just going to quickly skim this one about Jack Russell Terriers because you mentioned deafness. So this is why maybe in some forms of albinism, in some mammals, because they do say that this phenomenon they don't say is necessarily conserved in mammals, but they say the site is conserved in mammals. Is that what they're saying? That is highly conserved. The site is highly conserved, or the retrotransposition is highly conserved? I think they're saying the site is. Regardless. So this is albinism and a disease. So let's see what this, and by disease I mean cancer. And this one you pointed out was about deafness. So they're doing association between patterns of pigmentation and deafness in a dog with long documented history. So this is useful because as a sort of a analysis tool. So a breed is another good way of saying a really inbred organism. So sometimes in science, in some of our model organisms, we use groups called isogenic lines. And this basically means that they are so inbred that any one individual is not too different from any other individual. They're not clones, but there's very, very little to no genetic variability. That's easier in like flies, something small and simple. Mammals are way tougher, so we can't make completely inbred mammal lines. But, and actually the reason for that kind of comes out when you think about dog breeds, how some dog breeds have bad hips, some die really early, some, like apparently Jack Russell Terriers, have problems with deafness. So this is boring out because they have an accumulation of traits in this specific breed. So when you say inbred, you don't mean in the typical sense of them being related to one another, but you mean inbred as in they belong to the same breed. No, they are inbred in the first sense that you mean. They are more genetically related to each other than another type of dog. That becomes useful for scientists when you're talking about like an insect type of model organism. So we can just breed brothers and sisters together for many hundreds of generations, and they become so inbred that they are just super similar. And that's very useful for us because we can tease apart, mutants or differences as long as the rest of the background is all the same. The background being the rest of the genome. So humans are not considered genetically same because we are not a breed. Breed is just kind of a weird dog pet term. It doesn't really mean, as far as I know, it doesn't mean a whole lot genetically. Maybe it means a certain amount of inbreeding, or it means that you have a certain set of traits that are predefined. In terms of biology, no, it doesn't. I wouldn't say it has a strong meaning whatsoever. In biology, we talk about more relatedness. What percentage of the genome is similar between two individuals? Humans, we do not have breeds. We have like family groups, but most organisms don't have breeds. Breeds are an artificial construct for humans who like to categorize animals that we've domesticated. A more proper genetic way of saying that is simply that Jack Russell Terriers are highly inbred or are very genetically similar to other Jack Russell Terriers. So because of that high genetic similarity, they accumulate certain diseases because they don't have a sort of background mix to sort of dilute that away. This is hard to explain because I don't actually know what I'm talking about. Like your question, maybe I didn't answer your question fully. If you have thoughts about it, just put it down in chat and I'll try to answer. Okay. So this present study is intended to evaluate the heritability of deafness in Jack Russell Terriers, characterize the mode of inheritance, and evaluate the existence of a sex coat color or coat texture influence on the expression of sensorineural deafness. So basically they're asking, when we're breeding for these different characteristics, like outward characteristics that are useful in breeds, which, are just what the animal looks like to us, is there an explanation for why also deafness is increasing? By the way, these are the search queries that I've been using. I see. Okay. Yeah. And then I just see the search terms that you've used. Albinism, deaf. I see. Okay. I'm just going to grab these for later. I'm tackling this dog one for a second, though. Okay, so basically in this paper, it sounds like they're trying to figure out, we have a breed. And again, a breed is something that doesn't really mean much genetically in terms of nature, but is a useful term for humans who like to keep very similar animals. So like horses that are all brown and a certain height, or in this case, dogs that look a certain way. In this way, they look like a Jack Russell Terrier, which I think is a small little dog. Hold on. I spelled that wrong. There they are. Okay, so, a breed of dog that is called a Jack Russell Terrier is clearly small dogs with a certain shape head, certain floppiness to their ears, with a certain coat pattern. That's all the breed means. Anyways, when they're trying to get that breed, why is it that a lot of them have deafness? So there appears to be an influence of coat color on the expression of deafness. In an attempt to characterize the mode of inheritance of deafness in JRT, Jack Russell Terrier, a model of a single locus. A model of a single locus with a large effect on hearing is not supported by this data. I see. Further study is needed to determine. So basically they were wondering, if you get rid of or if you have a gene that codes only for the Jack Russell Terrier coat patterning, does that also cause deafness? Sounds like they're basically saying, actually, it's more complicated than that. It's weird because for humans, we typically use the term inbred to refer to people who are the offspring of people closely related to one another. That is true. That is the definition of inbred. If a Egyptian only breeds with Egyptians, that might be a breed. Like when we're talking inbred, I took inbred as sticking with certain type of subclass. Pitbull with pitbull, Jack Terrier with Jack Terrier. Might be inbred that you mentioned, but humans can do that too, in my head anyways. I see. So first of all, with the human classification of races within ourselves, races don't really exist genetically. They are more of a social construct. There are certain physical characteristics that people point out. So people of a certain lineage have eye folds, and as those people moved from parts of Asia farther east and then eventually into the Americas. Also, people near the equator, regardless of the continent, generally have more melanin production in their skin, so that causes different shades of color. Likewise, people more near the north, I think, the northern latitudes, have much less melanin, have a lighter skin color, and also like different shades of hair. So we are all saying sort of the right thing, but I guess I want to make sure we get this definition down. So hold off on these thoughts for a second. Let me get rid of my beautiful pictures. Defested, I gotta jump off. Thanks for a more formative stream. Send me any questions you mentioned earlier, e.g. warp drive. Absolutely. Thanks for watching. And I will let you know if I ever start up that Discord, just to honestly, as a list to collect things or ideas. All right. So, inbreeding. Both of you are saying the same thing. The first thing I want to get through, though, is that humans don't have what we would define as breeds, because humans are really similar to each other. Sure, we have different skin colors and maybe eye shapes. That's kind of it. We're really close to each other in the ways that matter, I guess is what I should say. If you think about the variability in different breeds of dog, you have Chihuahuas on one side of the spectrum, and then you have like Alaskan Malamutes on the other side. They're both dogs. That's why we don't really use the term breed, and I might just stop trying to say that word because it's just probably more confusing. So inbreeding is really a simple concept. So here we have in circles, we have female, in squares we have male. They pair up and they have children. The first level of inbreeding is, when they have two siblings, or excuse me, two children who are siblings, and then these two pair up and have a child. So this child has way more DNA in common than if via another lineage this male had a child with that individual, right? And if we look at the similarities between, and this is one way to measure inbreeding, if we just look at this grandchild and look at the grandparents, this grandchild, look at the grandparents, who is more inbred? Well, it's this one. Because this one had a higher proportion of genes come from both of these grandparents because the parents are siblings. This one over here, I'm just going to say not inbred, they have a whole other set of grandparents who are very likely genetically dissimilar. So inbreeding happens in humans to a degree, right? Certain communities, maybe first cousins will have children together. But generally, just because of human social constructs, inbreeding is really, really looked down upon across cultures. Maybe second cousins, but even that genetically is not so bad. So we as a group, even within sort of the groups that you defined, are not really inbred. When we're talking about like dogs being inbred, this is what we're talking about. We have one Jack Russell Terrier, another Jack Russell Terrier, and they have certain coat colors, and we really like those coat colors. So if they have two puppies, one thing that you might do is you might breed those puppies together. And then you might breed these dogs together. And then this other, this male dog way up here, he might have had, how do I draw this? He might have had a brother, right? Who was mated and had some children of his, or some puppies of his own. And eventually you breed these two together, right? So we're always crossing back, going back and forth, back and forth. And this goes on for many generations. This is how dog breeds were originally sort of defined. It's small communities of people who are looking at their dogs saying like, okay, this one community, they want medium-sized, blonde, shaggy dogs who like water and hunting. And after many, 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 many years of inbreeding over and over again, siblings, that's kind of extreme, but cousins to cousins, absolutely. Suddenly you get dog breeds like golden retrievers or Dalmatians or Jack Russell Terriers. So while every once in a while a human family might show up that is very inbred, that does happen. It's not to the same degree as domesticated animals. And that's mostly because humans control the breeding of domesticated animals. But when it comes to humans with other humans, we generally have the choice and we choose not to inbreed. And even within populations, that's not inbreeding. Inbreeding is, I don't know if I can say it's a short time scale, but it's much, much closer family groups. I guess I was just confused because you used the term breed when it came to inbreeding. So I thought you were implying that if two Jack Russell Terriers had a child with each other, even if they didn't come from the same lineage, they would technically be inbreeding. I see. Yeah, I was just improperly using that. They are definitely, a Jack Russell Terrier is more inbred than a wild wolf, but you're totally right. There's still larger family groups going on who are going to be more distant related, and it's not the same as breeding two Jack Russell Terrier siblings together. I see why I made you confused. So that's my fault. But thanks for asking the questions. All right, I'm gonna go back to this. So let me open this paper. So it seems like, unfortunately, this group actually didn't find a specific gene that was the inbreeding gene. Oh, very cute. Let's see what we have here. Yep, they're tracking a bunch of, so this is a pedigree, so just like what I drew, right there, looking at these different dogs up here, they're tracking the puppies that come out and finding the ones who go deaf. Interesting. And then they're also looking at their phenotypes, so auditory status, coat texture, coat color, and they're tracking males and females. So basically they're trying to see, like, hey, as a proportion, is there more likely to be deafness if they have a certain trait? So let's just say, for example, that Jack Russell Terriers had another coat color, and that coat color was purple, and 50% of every purple Jack Russell Terrier was deaf. Well, then you might think to yourself, oh, the genetic component might be the same genetic component as the one that makes them purple. But even so, Jack Russell Terriers don't have great genes. As a breed of dogs, I'm not sure. It sounds like if they're having a problem with deafness, probably not. Most pure breeds just don't have even great genes as a phrase is kind of hard to say. They are probably not very diverse in their set of genes. So I don't want to say whether or not they do or don't have great genes. But often that's why you see mutts being healthier than pure breeds, because they have a greater mix of different genes. And generally that means that if they have a bad gene from one, a bad gene from the other, well, they actually have a copy of good genes from the opposite. So I don't want to say anything about Jack Russell Terriers as a dog breed. I don't really know much about them. So, okay, so then okay, I see. Okay. So they're basically doing a bunch of statistics, trying to figure out whether or not there's effectively any component that they can point to and say, this heritable trait, which is usually some outward physical appearance, that is clearly linked to deafness. I'm just skimming over this. Frequency of estimated genetic parameter from Gibbs sample for heritability of deafness. Okay. Deafness, heritability of coat color, genetic correlation between deafness and coat color. Yeah, this is very genetics-y. Yeah. They basically say they couldn't figure it out. Well, that's too bad. I'm just reading this. Yeah, so basically they do find that there is heritability, if I'm reading this correctly, which obviously I've just been skimming it. If it seems like they did find that deafness is a heritable set of traits. Sorry, just turning off my microscope. Deafness is heritable in Jack Russell Terriers, but at least with the traits that they were looking at, it doesn't seem to be a one-to-one correlation. I just had a fun idea. It would be cool for you to go through some papers like these and show us when they're implemented proper research techniques and where they've improperly used statistics/research. I would love to do that. Certainly others on Twitch who kind of do the science thing also do that. So maybe I should just get my button gear and really make that Discord. It's easier to be shown a paper and just asked about it. But yeah, that definitely sounds like something to do on Sundays, the office hour Sundays. So you know what? I've gone over the time that I meant to, so I am going to wrap it up. But to your point, Slayer Darth, because I think it is a good idea, I think I'll take some of the papers that you sent and some of the search terms that you sent. I'll teach myself some more about albinism so we can kind of return to this. And why don't we talk about some of these papers next Sunday? I think that will be a lot of fun. Yeah, no. Don't apologize for giving me more resources and more ideas. At the moment, let me see if it works. Hold on. Discord. Yeah, so currently I don't have a Discord. I will make one and I will put it on my channel page. So if you want to join, you can, and just send me stuff. Otherwise, we can talk next Sunday about some more about this albinism and some of these papers, and I can walk a little bit more clearly through it, so I'm not surprised by the themes. But otherwise, on Thursday, I plan on doing a stream talking about the science of sci-fi. We're going to be talking about cloning. We're going to be talking about the TV show Stargate SG-1, and we're going to have a good time picking apart some of that stuff. So thank you, everyone who's been participating and asking great questions. Thanks for all the new follows, and I hope to see people next week. So thank you. Oh, if I have a Discord account. Yes, I have a Discord account. I can invite you to some similarly themed streamers Discord channels. I'm on a lot of these Discord channels, but yeah, no, if you, once I start this and you join in, invite me. I'm happy to have more places to advertise. So that's a great idea, so thank you. Yeah, I'm not going to share my Discord. I think I use my like personal Discord for everything, so I'm going to double check that before I give out my Discord name, but we'll figure that out later. Okay. But anyways, I just want to thank everyone for watching, and I'll hopefully see people next week.